In the past few years, a drug-resistant type of bacteria commonly found in hospitals has evolved to use an antimicrobial weapon against its own kind, wiping out competing strains and becoming the dominant version. Researchers at the University of Pittsburgh School of Medicine made this discovery while analyzing local hospital data, and then confirmed it's happening worldwide. This finding, recently published in Nature Microbiology, could help shape new treatments for some of the world's deadliest bacteria. It also highlights a powerful system developed at Pitt and UPMC that combines genetic sequencing with AI-driven analysis to quickly detect and track infectious disease outbreaks. According to Dr. Daria Tyne, an infectious disease expert at Pitt, their lab closely monitors pathogens circulating in hospitals. When they stepped back and looked at the bigger picture, they saw a major shift happening with one particularly tough to treat bacterium. That bacterium is vancomycin-resistant Enterococcus faecium, or VREFM. As the name suggests, it's resistant to vancomycin, a widely used antibiotic. VREFM is especially dangerous for hospitalized and immunocompromised patients because it thrives when antibiotics wipe out other gut bacteria that would normally keep it in check. Infections caused by VREFM have a high fatality rate, about 40%, making it a very serious threat. Emma Mills, a graduate student in Van Tyne's lab, used the hospital's Enhanced Detection System for Healthcare-Associated Transmission, or EDS HAT, to track how VREFM has evolved. This system normally helps doctors spot infection patterns and prevent outbreaks in real time, but Mills realized that it could also be used to study how these bacteria change over time. When she analyzed 710 VREFM samples collected over 6 years, she found something striking. Back in 2017, there were about 8 different strains of VREFM circulating in the hospital, all fairly evenly distributed. But by 2018, 2 strains had started to dominate. By the end of 2022, they accounted for 4 out of every 5 infections. So what gave these strains the upper hand? Mills discovered that they had developed the ability to produce a bacteriocin, essentially a bacteria weapon that kills or suppresses other bacteria. By eliminating competing strains, these dominant versions had free access to nutrients, making it easier for them to spread and take over. That raised another big question. Was this just happening at their hospital, or was it a global trend? No previous research had looked into this, so Mills turned to a public database containing over 15,000 VREFM genomes collected worldwide between 2002 and 2022. Sure enough, the same pattern was unfolding on a global scale. This was a surprising discovery. No one expected such a dramatic shift. Once these bacteriocin-producing strains enter a hospital, they outcompete other VREFM strains in a patient's gut, spreading very rapidly. For now, this doesn't seem to make infections any worse for patients, but it could have important implications for treatment. With fewer VREFM strains out there, researchers might be able to develop therapies targeting just 1 or 2 dominant types rather than a whole range of them. It also raises an interesting possibility. If bacteriocins are so powerful at eliminating other bacteria, could we harness them as a treatment? Instead of fighting these bacteria with traditional antibiotics, we might be able to turn their own weapons against them. That's it for today, and thank you for watching. And as always, have a Voltastik day.